Hello everyone. At first, I would like to introduce myself. My name is Takumi Tsutaya, and I'm researching the breastfeeding and weaning behavior in past human populations and extant great apes like orangutans and chimpanzees, like in Malaysia or in Uganda. But today, I will talk about the more ancient times. At first, I would like to introduce my research background. Under the BHAP and Core projects, we are excavating the Hamanaka 2 sites on Levin Island, Hokkaido, Japan. Hamanaka 2 site is here, and here we are. Because stick may crack the. So, we have already done several years of excavation, and multidisciplinary approaches have been applied to several materials. And this is the photo of excavation. Cultural sequences from Zoku-Jomon or Epi-Jomon periods to Okhotsk period and probably Ainu periods were discovered. And people subsisted here were maritime hunter-gatherer fishers. And there were many rich animal remains in good preservation. And already I have said, multidisciplinary approaches have already done for these materials. At first, I should talk about the overview of Hokkaido prehistory. Levin Island is here, and here is Hokkaido. And in this presentation, I will talk about two chronological periods, namely Zoku-Jomon and Okhotsk. Zoku-Jomon people was successors of Jomon people, Jomon hunter-gatherers. And Zoku-Jomon people subsisted Hokkaido Island, but about 6th to 10th century, Okhotsk people come down from more northern part of like Sakhalin or mainland Eurasia and go down to Hokkaido Islands and moving through these directions and go to Kuril Islands. And contemporary Satsumon and Okhotsk populations were emerged, or Okhotsk people were observed into the Satsumon populations, and later they were developed to historical peoples. And these figures indicate site distribution in prehistoric Hokkaido, and this is Zoku-Jomon period. Site was located all over the Hokkaido Islands, but in Okhotsk period, site distribution is strictly limited to coastal lines of northern eastern part of Hokkaido. And Levin Island is important because this is like cross-cultural sites of mainland Hokkaido and northern part of continents. So this is my object of my study, and what kind of food sources was used in Levin Island, because Levin Island is small island, and especially in winter, environment is very harsh. So terrestrial food sources are difficult to obtain. So what kind of food sources they were used? I adopt approaches of stable carbon and nitrogen isotope analysis. As you may know, stable carbon and nitrogen isotope ratios were differentiated by different type of food sources like C3 plants, aquatic foods, and marine products. And by analyzing and comparing the isotope ratios of food sources and humans, we can reconstruct past dietary habits of ancient human populations. After the several years of excavations, we have many biological materials from Hamanaka 2 site. And the number of used samples in this study is indicated here. And I extracted collagen from each bone and analyzed them by using elemental analyzer coupled isotope ratio mass spectrometry like this. Yeah. And this is the overall result of my analysis. All data points were plotted in these figures. And yeah, as a general rule in stable isotope ecosystem, carbon isotope ratios are higher in marine organisms than terrestrial organisms. And as a general rule, nitrogen isotope ratios are higher, approximately 4 per mil in consumers compared with prey. And nitrogen isotope ratios also indicate marine signals. And yeah, you can see these figures. Human is on the top consumer, and dogs here, marine mammals, marine fishes, and terrestrial mammals. At first, I would like to present the result of human, dogs, and pigs. This is another representation of the same data, and this is backplot. And as you can see, human is on the top level consumer in this population, and probably consumed marine mammals, marine fishes, and domestic animals. And the archaeologists working in this project reviewed many cut marks on dog skeletons from Hamanaka 2 site. And the isotope result is consistent with such evidence. And dog is terrestrial mammals, so dog should be around here because this is terrestrial range. But dogs indicate very strong marine signals. This indicates dogs probably consumed marine fishes and marine mammals as their main dietary sources. Because dogs cannot fish or hunt marine mammals, they are probably fed carefully by humans. And pigs is more diversified in their isotope ratios between terrestrial food sources and marine food sources. This may indicate mixed diet in pigs between terrestrial and marine food sources. And probably they were kept as semi-free ranging and not cared as like dogs. And this is isotopic differences by sub-period of Hamanaka 2 sites, like this. And this is Zoku-Jomon periods, and this is these are Okhotsk period. And dogs indicates no chronological differences in carbon and nitrogen isotope ratio. This result is consistent with morphological study. The archaeologists working in our project found no morphological changes in dog skeletons from Zoku-Jomon to Okhotsk period. Yeah, so diet is also not changing. But in pigs, maybe, yeah, I found no significant differences between sub-periods. But the oldest Okhotsk Poreds period, pigs might indicate lower carbon and nitrogen isotope ratios here, but sample size is small. Pig domestication in Levin Island was studied by zoo archaeologists, and Toada period is its initial introduction of pigs into Levin Island, and there was no breeding colony in Toada period. But after Kokumon or another Okhotsk period, pig breeding colony was established in Levin Island. So maybe diet was also differ between these sub-chronological periods. And yeah, final results. And yeah, we can also see the differences in several marine mammal species by using stable isotope ratios. As I have already explained, nitrogen isotope ratio is indicator of trophic level in ecosystem. Consumer indicates higher nitrogen isotope ratios. So plankton eaters like whales and dolphins indicates relatively lower nitrogen isotope ratios because plankton is at the lower level of trophic levels. But in fossils and sea lions, they consume marine fishes. Marine fish is at the higher trophic levels. They indicate higher nitrogen isotope ratios, and their consumers also indicate higher for seals and sea lions. Yeah, and there are also in species differences in seals. And this is the result of fox excavated from Hamanakatu site. And yeah, fox is terrestrial mammals, so they should be around here. But they are distributed around here in the marine range. This may indicate foxes consumed a significant amount of fish marine products in their lifetime. And it is also interesting. Fox indicates chronological differences because Okhotsk fox is around here and Zoku-Jomon fox around here. So older fox indicates relatively lower nitrogen isotope ratio. This may indicate, foxes scavenged human food debris because in Okhotsk period, people consumed more fish or marine products, and they discarded them, and thick fishbone layers were formed in Hamanakatu site in Okhotsk period, newer period. So, assuming foxes consumed human debris, this kind of result will be obtained. And isotope signatures in marine fish is also in interest. As a general rules, carbon and nitrogen isotope ratios differs in some species, such as open ocean material indicates lower carbon isotope ratios. So some kind of migrant fish sometimes go to the open ocean and they consume lower carbon, lower isotopic carbon. So herring is here, and they are migrant fishes, and they indicate relatively lower carbon isotope ratios, probably derived from open ocean. And in nitrogen isotope ratios, organisms from higher trophic levels indicates elevated nitrogen isotope ratios, and plankton eaters like herring hokke indicates herring hokke relatively lower nitrogen isotope ratios because plankton is at the lower trophic levels. But small fish eaters like rockfish, cods, flounder indicates higher nitrogen isotope ratios than plankton eaters. This is differences in trophic levels. And some fish species indicate chronological differences in their isotopic ratios. This is the result of nitrogen isotope ratios, and no chronological differences were found in carbon isotope ratios. This may indicate the fishing places was not changed between Zoku-Jomon and Okhotsk period, because carbon isotope ratios differ by sea regions or origin of water. So if the fishing places differ between periods, carbon isotope ratios of fished fishes differ. But in Hamanaka 2 site, we cannot find chronological differences in carbon isotope ratios. But in nitrogen isotope ratios, some fish species indicates chronological change, like cods and herrings. In older Zoku-Jomon periods, cods and herring indicates significantly higher nitrogen isotope ratios than more recent fishbone samples. This may indicate older Zoku-Jomon periods targeted more bigger fish than Okhotsk period, because bigger fish tend to consume more carnivorous diet, so they would indicate higher trophic levels than small same species. By analyzing stable isotope ratios of fished fish bones, we can reconstruct fishing strategy of ancient human populations. This is conclusion. We can find feeding differentiations between humans and domesticated animals. This may alleviate the feeding competition in small islands like Levin in Hamanaka 2 sites. And isotopic technologies can detect species differences in several categories of organisms, and also sometimes relationship between animals and humans, and like fishing strategies in humans. Thank you so much.